Welcome back, students, to the part 4 of our video series on DNA. Earlier we had explained upon the bacterial conjugation experiments which were being conducted by Tatum and Lederberg, and now let's continue with the experiments which were being carried out by Hershey and Chase to explain how a virus could transfer its genetic material into DNA. So this Hershey and Chase, now they are Alfred Hershey and Martha Chase in 1952. They conducted this series of experiments to prove that DNA was a genetic material in the case of viruses. And the virus that they used here is a T2 bacteriophage which could infect an E. coli bacteria. And for this, they grew the phage in one of the two isotopic mediums in order to radioactively label a specific viral component. So let's go into an animated video which would help you better understand the experiment. So this is Alfred Hershey while Lederberg was doing his work on bacterial genetics. A group of others, including Hershey, was doing some experiments at the Cold Spring Harbor Laboratory, which is hosting this animation itself. They were conducting some experiments on bacteriophage genetics. And as we had mentioned earlier, bacteriophage is a phage for a short, and a virus is that especially attack and infect bacteria. This is a T4 bacteriophage, and you can see the picture. It is having a capsid as well as some tail fibers. So the phage had an outer protein coat and an inner core of DNA. The outer protein coat as well as the DNA, and you can see the tail fibers which have been present in them. The phage usually rely on the bacteria to reproduce, and from electron micrograph studies during infection, it was been found that the phage attacked to the bacteria using their tails. And after attacking, the genes are pumped into the bacterial host, which then direct the bacterial enzymes to replicate new phage particles. This is quite evident in the animation which they have provided. Now they need to find out what exactly caused the transformation of the bacteria into the phage producing factory. Could it be that, as Avery's work suggested, phage DNA was a transforming principle, and Hershey was curious, and along with one of his colleagues called Martha and Chase, they decided to test these ideas. So from a previous chemical analysis, they had found that DNA is highly rich in phosphorus and it has no sulfur. And it has also been found that proteins contain sulfur atoms but no phosphorus. So knowing this, they used the radioactive phosphorus and sulfur and to selectively label the phage DNA as well as the protein. And they designed an experiment to test which component entered the bacterium for infection. So there was a tube in which there was phosphorus 32 was provided, and in such a way that the coat, not the coat, the DNA, when DNA synthesis was being carried out by the phage, the phosphorus 32 would be incorporated into its DNA. And in another set of experiments, it was being seen that sulfur 35 was being provided, a radioactive sulfur 35 was being provided in the experimental tool, and since sulfur is a part of the coat, it would only be found in the radioactivity will be detected in the coat of the phage. And in the two parallel experiments, they combined the radio-labeled phage with bacteria that were not labeled, and they wait for long enough for the phages to attach, and then they disrupted the attachment by mixing the culture in a warring blender. Next, what they did, they spun the samples in a centrifuge to separate the phage from the bacteria, And because the bacteria are larger and heavier than the phage, the bacteria could collect at the bottom of the test tube as a pellet, and since phage has been found to be very small, it would stay in the suspension in the supernatant. So that's what you have, the pellet where the bacteria is being found, as well as the phage particles would be found in the supernatant. So in one of these samples you could have phosphorus 32 laid phages, and in the other one you could find phage particles which were being coated with S35. Now let's just examine the results. In S35 labeled the suspended phage and not the we could see that S35 was mainly found in the suspended phage particles but not in the pellet of the bacteria. And the new phage made by these infected bacteria did not contain radioactive sulfur. Therefore, the phage coat, which is made up of protein, was not used inside the bacteria to make new phage particles. Now, in the case of the phosphorus 32 samples, you can find that phosphorus 32 was always pelleted with the bacteria. That is, where is the bacteria? The bacteria was being found here. This is the bacteria. So on the bacteria you can see the phosphorus 32, and the new phage particles made from these infected bacteria contained radioactive phosphorus 32. So this indicated that phage DNA was used inside the bacteria to make the new phage particles. Now this is a view of the bacteria, that is phosphorus 32 is incorporated into the cells within the pellet of the bacterial cells. The phage coat is just a package that delivers the phage DNA into the bacteria, and they concluded that the phage DNA alone carries the instructions needed for the replication phages inside the bacteria. So DNA is a genetic material. You can see the DNA was incorporated into the cell, and using that DNA, more phages were being released out. So T4 bacteriophage is attaching onto E. coli in this pic. I hope this is clear to you. So with this we come to an end of the Hershey-Chase experiment. Let's see with each other into the next experiments of DNA. Thank you.